Hello everyone. Let's get started. Today we're going to be talking about prokaryotic cells. We've been talking about cells very generally, and today we're going to focus in on a particular category of cells, the prokaryotic cells. Remember, a cell is essentially a membrane-bound compartment within a living organism. It contains all of the molecules that are necessary to sustain life. The cell theory tells us that all living things are made of cells, and that all cells come from pre-existing cells through the process of cell division. We know through evolutionary biology that there had to have been an original cellular life form on planet Earth, and that all the other cells that have emerged since that time had to have come from that first cell. Of course, cellular life has changed dramatically since life first appeared on the planet, and we can arrange all of the different life forms into one of three domains of living things on planet Earth. We've talked about these domains. They include the bacteria. That was the domain that emerged first. The archaea, which emerged next. And finally, the eukarya. There's cellular life in all three domains. But there's a difference in that the bacteria and the archaea are single-celled life forms. The cell is the entire organism. There are also single-celled eukarya, but in the eukaryotic domain, we also have multicellular organisms, like humans. The three domains are arranged on this slide in a phylogenetic tree. Remember, we draw trees like this because it allows us to see not only the evolutionary history, but also how different organisms emerged in relation to each other across relative amounts of time. Remember, we interpret the base of the trunk of the tree as representing that first original cellular life form that emerged on planet Earth. Everywhere that you see a branching event, like here or here, everywhere that one branch on the tree splits into two or more, that's what we call a node, and that represents some event occurring in the environment, in the world, causing that one group of organisms to split into two or more groups of organisms, headed down their own evolutionary path. You can see that as the original life forms survived on the planet for some period of time, an event occurred that caused the bacteria to set out on their own evolutionary path. Drawing evolutionary history in a phylogenetic tree allows us to see, relatively speaking, when these branching events occurred relative to each other. In other words, the branches that come off first were the events that occurred farthest back in history, and the branches that emerge most recently occurred most recently. We read the tops or the tips of these branches as being our current understanding of where these organisms are in their evolutionary history. So we can't know precisely the day and time that the first bacterial organism appeared on the planet, but we do know, relatively speaking, some important information about our evolutionary history. For example, our current understanding is that the universe formed approximately 14 billion years ago. That was the time of the Big Bang. The Earth, in comparison, was not formed until about 4.6 billion years ago. That first cellular life, the first cellular life that appeared on the planet, 3.8 to 4 billion years ago. Now I've got an asterisk next to this number because this number has recently changed. Over the past several years, some new evidence has emerged. We used to think that the first life form appeared on the planet about 3.5 billion years ago. But some newly discovered fossil evidence suggests that it was a little bit earlier than that, 3.8 to 4 billion years ago. It's an important reminder to us that our evolutionary history is a work in progress. We construct our phylogenetic trees with the evidence that we have currently. And as we discover more and more and more evidence, we can make adjustments to the trees as needed. Now, this chart lists the oxidation of the Earth at 2.5 billion years ago. This should actually read oxygenation. The oxygenation of the atmosphere around the planet occurred about 2.5 billion years ago. There was a single-celled photosynthetic type of bacteria that evolved around that time called cyanobacteria. And the cyanobacteria, through the process of photosynthesis, released oxygen into the atmosphere and helped produce the oxygenated atmosphere that we still have today. Eukaryotic cells, we believe, first appeared around 2 billion years ago, but it was not for another additional billion plus years that those eukaryotic cells evolved into multicellular organisms. Remember, becoming multicellular was advantageous for these organisms because what it allowed them to do was compartmentalize the kind of work that gets done in a cell every day. Prior to that time, both prokaryotic and eukaryotic organisms were single-celled, and that meant that every single thing that had to be done every day to survive had to be done by that one cell. Once life forms became multicellular, they were able to compartmentalize jobs. In other words, some cells could be devoted to some of the work that needed to be done, and other cells could take care of other types of work that needed to be done. Multicellular organisms appeared about 900 million years ago, which is really not that long when you consider the age of the universe and the age of the Earth. The last two numbers on the chart are really more just interesting to us. They don't relate to prokaryotic organisms, but humanoids diverged from the other apes about 6 million years ago. And Homo sapiens, that's us, first appeared about 200,000 years ago. So we are on this planet for only a very short amount of time so far. I want to point out that Homo sapiens is the scientific name for our species. Remember, the first part of the name is called the genus. The genus name of an organism always gets a capital letter. The second part of the name is the species, and the species name always gets a lowercase letter. So our genus and species is Homo sapiens. So cellular life has been around on this planet for billions of years, and over those years it has evolved again into the three domains that we have now. Two of those domains, the bacteria and the archaea, contain single-celled prokaryotic life forms. Remember, a prokaryotic cell has no organelles in it and only a very primitive sort of pre-cytoskeleton within it. Eukaryotic cells are different. The cells that are in the domain eukarya have both organelles and a true cytoskeleton. So the two categories of cellular life are quite different from each other. By numbers only, the prokaryotes vastly outcompete the eukaryotes. There are many, many, many more types of prokaryotic organisms on our planet than there are eukaryotic organisms. When we look at the cells, the types of cells that you see in bacteria and archaea versus eukarya, one thing becomes very clear. There are many different types of cells in terms of their shape. We know that the shape relates to the function that a cell can perform, so it's not surprising that there are many different shapes of cells. What is interesting to note is that prokaryotic cells, again, the kinds of cells you find in the bacteria domain and the archaea domain, prokaryotic cells do come in different sizes and shapes, but they are not nearly as diverse as eukaryotic cells are. If you look at the image on the left-hand side of the slide, you can see shapes of bacterial cells. Note that these shapes are very similar to archaeal cells as well. We have this spherical shape here on the left. That's what we call a coccus. If you look all the way over to the right, you can see this elongated rod-shaped cell that's called a bacillus. In between those extremes, we have sort of an elongated or oval-shaped cell called a coccobacillus. And then we have a rod that's sort of bent or curved. That's referred to as a vibrio. Underneath, you can see two cell shapes that are vastly longer than any of the other cells and helical or twisted. These are the spirillum and the spirochete. These two cells can be difficult to differentiate. For our purposes, just know that a spirillum shape has many fewer twists, and a spirochete has many more twists. So there are a variety of shapes of cells that you find in both bacteria and archaea, but not nearly as diverse a set of shapes as you find in the eukarya domain. Remember, eukaryotes can be single-celled or multi-celled organisms. And in the multi-celled organisms, groups of cells are dedicated to different types of functions. So if you look at a cell, for example, like this one, this represents a nerve cell, or what we more correctly refer to as a neuron. This cell has these strange spider-like projections coming off of its surface. The membrane, in other words, is extended outward. This is a cell that has a job to do involving picking up signals from other cells and transferring that information onward to even more cells. These projections that are called dendrites are what pick up that information from the environment. It makes sense that the cell needs to extend its membrane in all different directions so that it's more efficient at picking up signaling from all parts of the environment around it. You can't see it in this drawing, but one of the extensions coming off of a neuron is going to be called an axon, and it's going to be greatly elongated, longer than all of the dendrites. While the dendrites receive incoming information, that information is then essentially summed up by the body of the cell and then sent along out the axon to the next cell that needs to receive that information. Neurons, like all cells in multicellular eukaryotic organisms, are highly specialized for the job they need to do. Muscle cells are another good example of this idea that the form of a cell is closely related to its function. Muscle cells, skeletal muscle cells in particular, have the job of contracting and relaxing. In other words, getting much shorter and then elongating. You can see that these cells have a very unusual shape. The cells themselves are elongated. We don't even call them cells. We refer to them as muscle fibers because of that elongated anatomy. Red blood cells have a dimple in the middle where the membrane is compressed inward. Remember, red blood cells do the job of gas exchange to the tissues. They give oxygen to tissues and they remove carbon dioxide. In order to do that job as efficiently as possible, these cells need to maximize their surface area. And that's what that dimpling does. In this area of the image, you can see what's called a guard cell on a green plant. These are cells that literally create these large pores in leaves that will open and close at different times of the day, allowing for more efficient collection of gas. Down here, we can see a single-celled eukaryotic organism called a Paramecium. As you can see, this cell has a particular shape to it, but it's not nearly as well adapted to function as these other cells are. And that's because a Paramecium is a single-celled organism. This one cell has to do all of the jobs that need to be done. It does have some interesting shape, though. You can see that at the sort of front of the organism, if you will. We have this more pointed shape to the cell. That's going to allow the cell to move forward through its watery environment more efficiently. And this little dipped and dimpled area right here, this is called the oral groove. This is where the organism can take in food particles, nutrition. This particular shape will cause the water, because of course it lives in the water, it's going to cause the water to sort of swirl and collect in this area, and then other organisms and molecules can be consumed through that oral groove. Form relates to function. The shape of cells relates to the job they're going to do. We see much less diversity of shape in the prokaryotic organisms, much more diversity of shape in the eukaryotic organisms. All of the things that we know about the size and the shape of cells in different life forms, we know because of the science of microscopy. We've been able to magnify cells to the point where we can see them for about 300 to 400 years now. In modern microscopy, the light microscope is the most commonly used type of microscopy. The light microscope can magnify cellular organisms and other types of materials from about 50 times to about 2,000 times. That's the current limit of magnification for a light microscope. A light microscope uses visible light in order to make that object on the slide visible to our eyes. It also uses a range of lenses, objective lenses, that help magnify the object. All cells can be visualized using a light microscope. It's just that some cells can be seen in greater detail than others. So prokaryotic and eukaryotic cells can be seen under a light microscope. It's just that the prokaryotic cells are smaller than the eukaryotic cells, so we see them in less detail. In order to see a prokaryotic cell in the greatest amount of detail, we have to use the highest level of magnification that we have available on a light microscope. That is, on most microscopes, about 1000x. Both the eyepieces on the microscope and that objective lens will provide some magnification. Eyepieces magnify about 10 times, and the highest magnification objective lens will magnify about 100 times, for a total of 1000x, or 1000 times magnification. This high-powered lens on a light microscope that can magnify 100 times is called an oil immersion lens. You literally have to put a drop of oil onto the slide over the specimen in order for the lens to show that specimen through the eyepieces. Modern light microscopes have multiple objective lenses available on them, and they also offer 3 different viewing options, depending on the kinds of cells that you're trying to visualize. Those viewing options include what's called bright field, dark field, and phase contrast. Now, this term field just refers to the background. In other words, there are cells or other objects on a glass slide, and then there's a background around those cells. So, in bright field mode, the background or the field behind the cell is going to be brighter than the cell itself. In dark field mode, the opposite is going to be true. The background is going to be darker, and the cell will be lighter. This image down here on the left, this shows cells that were being shown magnified using the bright field option. You can see that these cells, there's one whole cell and then a portion of another cell. These cells have been stained. We commonly stain cells when we're examining them under a microscope because the stain is going to help us see them more clearly. You can see on this particular image that the cells are darker than the background. This is bright field microscopy. Now compare that to this image over on the right. This was taken using the dark field option. So the background or the field is almost black, and the cell here is much lighter or brighter. There's also some debris visible on this slide. When we're examining cells, particularly cells that are coming out of the natural environment, sometimes we pick up little bits of debris in our sample, and you will see those under the microscope. Now, the final option that we have available to us on a modern light microscope is phase contrast. This is a eukaryotic cell visualized under phase contrast. You don't really see much of a difference between the cell itself and the background in terms of one being lighter or darker than the other. But what you can see is more 3-dimensional detail, more contrast between the different portions, the different layers, if you will, of the object versus its background. Phase contrast is particularly helpful when we want to examine a living cell under the microscope. We want to look at a cell while it's alive. Sometimes we want to know if the cell can move under its own power. Sometimes we just want to see it alive so we can see how it looks in terms of a living organism. Remember, most of the time we stain cells, and stain actually kills a living cell. So if we don't want to stain it, we want to keep it alive, phase contrast is a nice option for viewing that unstained cell. There are other kinds of microscopy in addition to light microscopy. One type is called electron microscopy. An electron microscope can give us the greatest amount of magnification for cells that we currently have available. An electron microscope can magnify up to 10 million times. Electron microscopes don't use visible light in order to show the specimen on the slide. Instead, they use a beam of electrons. And that's going to allow not only for greater magnification, much greater magnification, but also much higher resolution. In other words, the image is going to show the object much more clearly to our eyes compared to the background. There are two kinds of electron microscopy that we need to be familiar with. One is called scanning EM, and the other is transmission EM. We abbreviate those as SEM and TEM. Now, the difference between these two kinds of electron microscopy is that scanning EM is going to show us the surface details of a cell, and transmission EM is going to show us the internal parts of the cell, the details of the inside of the cell. One thing that we need to develop a feeling for as we learn more about different types of cells, we need to develop an understanding of the size of cells in relation to each other. Sometimes it's difficult to imagine, for example, how large a bacterial cell is in comparison to an animal cell. What you see on this slide is a chart that makes comparisons between the different sizes, the relative sizes of different cell types. Here is an animal cell right here, and you'll notice that the animal cell measures about 10 micrometers. The scale on this image, this diagram, runs from 1 millimeter off to the right, all the way down into the nanometer range. Remember that 1 millimeter has 1,000 micrometers in it, and 1 micrometer has 1,000 nanometers in it. So we're traveling across a distance here in terms of measurement that varies between 1 and 1,000,000. There are 1,000,000 nanometers in 1 millimeter. So animal cells tend to range in the tens of micrometers in size. So 10, 20, 30 micrometers approximately, depending on the type of cell. When you compare that to plant cells, again, on average, are larger. Plant cells tend to be about 10 times larger than animal cells. That's what these numbers are telling us. On the opposite side, we can see that bacterial cells tend to be about 10 times smaller. The average bacterium, if there is such a thing, is about 1 or 2 micrometers. Now, If you go down below the scale, you can see these sort of light pink bars drawn in. There's one for the light microscope, and there's another for the electron microscope. I want you to make note of the fact that bacterial cells, and this would also apply to archaeal cells, bacterial and archaeal cells, these prokaryotic cells, they are at the very edge of what can be seen with a light microscope. So yes, all cells can be visualized with a light microscope. The smallest type of cell that can be seen is a bacterial cell or an archaeal cell. Viruses cannot be seen with a light microscope. Viruses are not cells, remember. They don't have the characteristics that cells have. Viruses fall in the nanometer range of size. The average virus runs from anywhere from tens of nanometers up into the hundreds of nanometer range. We can see viruses with electron microscopes, but we cannot see them with a light microscope. The next couple of slides in this slide set we're not going to go through, but I included them just in case you need to review this particular material for yourself. The next couple of slides talk through how to do a unit conversion using the kinds of metric units that we encounter a lot when we're talking about things like cells. Most commonly, we use units including millimeters and micrometers or micrometers, and nanometers. We're talking about small units of length here. Sometimes we need to convert a measurement from one unit to another, and we do that using a process called a unit conversion. Again, I'm not going to walk through how we perform a unit conversion. Hopefully you've learned that technique at some point in your education. It's a very important technique to know, though, not just for measuring length, but also measuring volume and measuring weight. As you know, we often have to convert from English units to metric units, particularly in science. And being able to do that in a systematic way is an important skill to have if you plan on working eventually in the field of science. So we're not going to talk specifically about how to do a unit conversion, but I included those slides in case you feel you might need to review that for yourself. What we are going to spend a couple minutes talking about is the idea of the size of a cell being limited in nature. So we just saw when we looked at the scale on a previous slide that cells range from the single micrometer unit upwards into the hundreds of micrometers. That's a vast size difference, but relatively speaking, it's not a particularly large range in size. And sometimes it's an interesting question to ask, why is it that cells are the size they are? Why haven't they evolved to be bigger, for example? The answer has to do with surface area and volume, and how those two things relate to each other in a living cell. It turns out that cells that have a high surface area to volume ratio have an advantage. In other words, that's an adaptation that helps a cell survive. A high surface area to volume ratio, in other words, a high surface area compared to its volume, is an advantage for a living cell for a number of different reasons. Cells that have this ratio are going to have a more rapid exchange of nutrients and wastes across the membrane. Remember, a living cell has to constantly adjust its living environment by taking in nutrients and expelling waste material across its membrane. So it makes sense that it needs a good amount of surface area on that membrane in order to achieve this exchange most efficiently. But there's a limit. There's a limit to how much surface area it can have in relation to its volume. Cells with a high surface area to volume ratio are going to grow faster. Cells that have this ratio are also going to mutate faster. Now you might say to yourself, well, gosh, I thought mutation was a bad thing or a dangerous thing for a cell. Sometimes that is true. Sometimes a mutation, or a change in the order of the bases in the DNA molecule, sometimes a mutation can cause problems for a cell. But there are other times when mutations give cells new traits, traits that are actually adaptive, traits that make the organism better able to survive. Cells with a high surface area to volume ratio have a faster or higher mutation rate, and therefore they evolve more rapidly compared to cells with a lower ratio. And this rapid evolution, of course, is advantageous to that particular type of organism. All right, we're going to switch gears now and talk more specifically about the details of prokaryotic cells.